This is the second part of lecture 19. I made a few changes, which is why I didn't do the recording quite as promptly as I should have. And I'll spend this part talking, just introducing the life cycle of the mouse, so the general processes, and we'll get sort of up to, in today's prep in lecture 19, we'll get up to sort of this implantation stage and this kind of unusual looking embryo, which is referred to as the egg cylinder because of the shape. There are some key features. Mice are a very good model organism for mammalian development, but it's interesting that there's a couple of aspects of development where humans and the chick are actually more similar, and that's about the shape and sort of in this stage and through the gastrulation process. So I'll be talking about those towards the end of this lecture and the beginning of the next one. So after the egg is fertilized, of course, there's cleavage that takes place, a series of developments. By the time we get to this blastocyst stage, we've got a fluid-filled cavity here. We've got the inner cell mass, and we've got cells around the outside. So we already know that a certain batch of the cells inside this inner cell mass are going to form the full embryo. And for mammals, which are placental mammals, the female nurtures and provides oxygen to the embryos while they're developing inside her body. So there's a lot of extra embryonic membranes and tissues. So the placenta gets formed, which is a combination of maternal and embryonic tissues. But the original implantation into the uterine wall comes from the embryo, right? Yeah, and the formation of the umbilical cord and other cells that give rise to those extra tissues. And during the cleavage stages, there is a process called compaction, which is required for the process of cavitation, which is the formation of this blastocele, this fluid-filled cavity. And so we'll be talking about that and how cell adhesion molecules impact that, because the proper adhesion, the proper connections between cells makes a big difference to lots of things. I've been talking recently about how those connections could lead to epibole, right, in one of the previous preps, and how changing those adhesions allow sheets of cells to move away from the sheet above them, and so on. So cell adhesion and the connections between cells are really important in how they behave, whether they move or not. And if they move, do they move as a group or individually, and that sort of thing. So something to kind of keep your attention on. Yeah, and so these, I've just got them listed on the side here. These are unique features of the mammalian blastocyst, and today we'll be talking about all of these. And then next time I'll be talking about gastrulation. It's sort of here and through the gastrulation process and the turning that the rodent embryo was a bit different from humans. And so I'm going to talk about this in lecture 20, and then I will introduce just a little bit about human development. I think students are usually most interested in that. Okay, so as mentioned, rodents are a good model system, and especially the mouse, and it's very well studied. It's a genetic model organism as well as developmental. They have a really short life cycle. It's just a few months to adulthood, and so compared to humans, it takes 9 months just to grow the baby, and then years and years before maturity is reached, and so on. So a very slow life cycle. And so when we're looking at human development, it's possible to look at early stage human embryos because people do get in vitro fertilization and so on, and the embryos can be produced in a dish, but it's extremely limited, not for technical, but more for ethical reasons. Playing around with human embryos there's a lot of issues, right, with those. Currently, there's a lot more work done because there's less ethical questions around using different kinds of stem cells and understanding how those stem cells um, are modified in order to promote the formation of certain structures and so on. I've probably mentioned this a couple of times because I have great admiration for Lisa Julian, who's currently in the MBB department, but she was in biology last year, and she teaches a stem cell course, and she's kind of a rising star. They produce little brain organoids in dishes and so on, and so there's lots of cool things you can do with stem cells, and this means you don't have to use human embryos to study those processes. So this really helps, not just because it's easier to work with these cells in dishes and so on, but because you're not disrupting an actual sort of embryo, which many people have real problems with. Um, and then historically, and we still do this, I mean, when we're trying to figure out how things work in one organism, we often use the other model systems that we have because, one thing you will be noticing, Notch does similar things in lots of different embryos. Um, Dorsal ventral signaling in invertebrates is practically the same as in vertebrates, except that it's upside down. So dorsal versus ventral are reversed, but the molecules tend to be the same, and the kinds of signaling that goes on tend to be the same. There is a lot we can learn about ourselves simply by having a look at some other model organisms. So the 2-cell stage, 4-cell stage, there's a certain pattern, which I'll talk about in a second, of division. Now, I talked when I spoke about sea urchin, the fact that cleavage is radial. In the chick, it's very what would you say? It's not along any particular axis. It's the cells above the yolk versus sort of closer to the yolk. But in this embryo, we have what is called rotational cleavage, and I'll talk about that, I think, on the next slide. There's pretty much no yolk, or if there is, it's a very tiny amount, just enough to sustain the embryo for a few days, 4.5 or so days before implantation. And so we go through cell division. We get to the 8-cell stage. This is the morula, a solid ball of cells. But there is a process here referred to as compaction. And when you look at the pre-compaction and post-compaction, when you look at it, it's compacted to the extent that it's really hard to see. Like, you could just barely make out the little divisions between the cells. So what's happened is the cells they're stuck to each other here, but they're stuck to each other along a wider surface, and they've gotten really close together, and much of the surface of a particular cell, except for the part on the outside, is connected to the other cells, right? So I know it's not that easy to see. I'm just outlining just a little bit of kind of how those cells are organized. And this is absolutely necessary. What happens is a watertight seal is formed on the apical surface. So if you look at a cell, 2 cells that are next to each other, there's a number of different junctions, but if a cell has an apical and a basal, which they do, then we tend to find the watertight connections between them up on the apical surface. And then there's other kinds of connections. There's lots of different like about 4 different kinds of junctions, and they're composed of a variety of different combinations of molecules. But key is making sure that these cells are connected very tightly wherever their surfaces meet, and that's a prerequisite for then the influx of salt and water that generates this cavity inside the ball of cells. Okay. Yeah, and so around this stage, we will start to see implantation. and that means that the embryo, some of the cells are responsible for starting that burrowing right into the uterine wall, and then the embryo sort of develops in there, surrounded by the uterine wall. And most of the images that you're going to see are not going to show that, because it makes it too hard to interpret. So we're working with schematics for the most part. So for the echinoderm, remember, I mentioned that we have radial cleavage. And what that means is that all the cells divide on the same plane, and it is at right angles to the previous one. So the first one divides the cell into two cells, and then the second one, which is this one, and it goes this way, they both divide with the same axis, and that gives us four cells. And then the next one is going to be here, which is also 90 degrees rotated compared to the previous one. And so we have these sort of, all the cells are dividing. This is only for the first few divisions, but in the sea urchin, all the cells are dividing on the, on the same plane in a coordinated series of divisions, and they're at right angles to each other. Interestingly, in mammals like the rabbit and the mouse, there is rotational cleavage. So what happens is the first cells divide to make two daughter cells, and then when this cell divides, the plane of cleavage goes in this direction, but when the other cell divides, the plane of cleavage goes in another direction. And so the plane of cleavage of this division and this division are at 90-degree angles with each other. And it's referred to as rotational because it's like if you were to take mentally these two cells and rotate them 90 degrees, then you'd have what looks like this, right? So it's and then if these cells were to divide again, then if these ones divided this way, then these ones would divide sort of that way, so that they'd be divided again, rotated 90 degrees with respect to the other cell. And again, this only applies to the first few divisions, because after that, different tissues start getting specified and they start dividing in their own way, not all synchronously, and then not necessarily with planes of cleavage rigidly specified. Okay, so that's the first part. You've got this rotational cleavage. After the 8-cell stage, we get what is called compaction. And so this is probably a better image than the one I showed before. It comes from this. It's quite a recent paper. And it shows that compaction. And what's really interesting about this diagram is you can almost see these little sort of connections between the two cells. So that's pre-compaction, and this is post-compaction, and you can see that, it's not that the cells aren't touching each other, they are, but when they're compacted, they sort of flatten down and more of their surface areas are in contact. And so the whole embryo looks a little bit smaller, and the cells are kind of flattened against each other. And we're basically maximizing the contact between them. Now the cells are drawn together first by adherens junctions. These are they have cadherin proteins, and then on the the most apical surfaces, which is in this embryo is on the outside. Along the outside are the tight junctions. Okay, and we're going to look at that. So this is just a sort of a schematic that just goes through the kinds of junctions. We're not going to talk about desmosomes, or there's hemidesmosomes and so on. And there's other kinds of molecules. But we're going to look at the adherens junctions. Adherens junctions sometimes include nectin proteins these are homotypic. So that means that a nectin molecule on one cell is connecting to the nectin molecule on another cell. They bind to each other this way. E-cadherins are the same. They are homotypic. That means the two of the same kind. So if you had E-cadherin here and B-cadherin, I can't remember what the other one is called, they wouldn't bind to each other. It has to be the same one on both sides. And we'll talk about cadherins more later when we talk about some other developmental processes in vertebrates. So that's one kind of junction. This kind of junction is important for holding cells together, and it's important because it can be found in multiple locations along a cell. But it's not watertight, so water molecules can move sort of through here and around here. And so what's really needed in order for cavitation to happen is you need to have tight junctions. And they're called tight junctions because they make an actual physical seal, and water cannot get through here. And so it's like a watertight seal. And so the molecules, you can't see them very well, but JAM, A, occludin, and claudin are the molecules that are responsible for this type of junction. And something else to just note is that many of these molecules associated with cell junctions, they're not just holding the cells together. They're connected also to cell shape because they are connected to actin filaments and the cytoskeleton. So there's a lot of proteins here. I'm not going to expect you to remember these, but just the idea that the molecules of the junction also connect into the cytoskeleton of a cell. Is that everything I wanted to tell you? I think that is everything I wanted to say. If you have any questions, of course, you can always ask. But importantly, adherens junctions, and then see how the tight junctions are shown here. You can see that molecules could move through here, but if there's water trying to get in or out, it's not able to pass through these junctions. And so the blastocyst is formed by cavitation, but that requires the compaction because you have these tight junctions that hold the cells together on the outside, which happens to be the apical regions of those cells. And then what happens is that water and salt are pumped in to the cells. The salt is pumped out into the center. And when that happens, the water just follows passively, because when you have high salt concentration, the water is drawn across. And so you wind up with this sort of fluid-filled, sort of salty cavity. And the water can't make its way out. It's pumped in, or it moves in through the cells because of the pumping of the salt. So, and then the water follows the salt, but it can't get out, and that's why the cavity is able to form and the fluid is able to fill up in there. And this just shows basically what I've said. You can have a look at that. Yeah. And then, so after a few more divisions, so this is early, this is 32 cell stage. By the time we get to the blastocyst stage, I think we have something like 1,000 cells. It's just quite a large number compared to this stage. So a few more divisions, and what happens is that there's cells inside. That's the other thing that happens during this early stage, is that there's divisions, and the divisions create outer cells and inner cells. The outer cells are going to be, they're going to contribute to the placenta and other extraembryonic membrane and structures. And then the inner part is the inner cell mass. And most of that is going to form the epiblast. So remember the chick embryo, it has the epiblast above the subgerminal space. And that's where the majority of the cells for the embryo are going to come from in the end, after gastrulation and everything. And the troph ectoderm, troph means feed, so troph, you can see why it's called that, because these are going to end up contributing to the placenta and the feeding and care of the embryo. And the blastocele cavity is here. So you've got these inner batch of cells, but they're offset. They're not sort of dead center. They're always off to one side. And the blastocele cavity is sort of there. Okay, so this is what we were left with. Um, Now, the inner cell mass will divide to form the epiblast, which is basically where the whole proper embryo comes from. So that's going to be the embryo, and the primitive endoderm, which is going to contribute to the extra embryonic membranes, and some of this becomes the yolk sac. There isn't much, if any, yolk, but a yolk sac still forms, and it comes from primitive endoderm. And then after, so implantation begins. And so the trophectoderm, which I didn't point out, the mural trophectoderm, which is down here, and which kind of forms all around here. And it's just a little bit up top here, that's the polar trophectoderm. I'm not really concerned that you memorize the difference between them. But if you remember that the trophectoderm, um, especially away from the polar region, will go through these. They're kind of like going through the cell cycle, but without actually dividing. So the DNA duplicates, and the cell gets ready to divide, but it doesn't divide. So, and that happens multiple times, like not just once or twice. So you end up with these things called giant cells, and they're giant because they keep duplicating and getting ready to divide, but they don't actually divide. So they're big cells, and they are part of the cells that sort of go into and invade the uterine wall to start attaching the embryo and to start forming the placenta. And then the polar cells, they make this ectoplacental cone, which is just part of, the burrowing in to the uterine wall. And this red line is just to sort of show you that this is kind of the uterine wall, and this part here is the embryo property, a proper the embryo proper. Okay, and there is endoderm lining this blastocele cavity. And the blastocele, so the blastocele with some of this endoderm around it is going to form a sort of yolk sac. And then the whole cylinder, this is referred to as the egg cylinder. And then this visceral, right here, this visceral endoderm is on the outside. So notice the color coding, because this is the epiblast, and this is going to form most of the embryo, and ectoderm and mesoderm. But the endoderm is kind of on the outside, and this is one of the peculiar things about mouse embryos, is that they develop as this little cup instead of a flat disc. And they kind of start off inside out with the endoderm on the outside and the others on the inside. And then there's this weird sort of turning process that occurs after gastrulation, which I'm actually not going to go into, because it's just that there's a weird twist, and then the whole thing flips around, and it's not easy to picture for anybody, including me. But there is this kind of inside out aspect to the embryo at this stage. And then by 6 days, you can also start to see the formation of this amniotic cavity. So you've got a little bit of a cavity here, and it gets bigger as we go on. It's just half a day later, and it's something that might be counterintuitive. So we just talked about forming a cavity by sealing the embryo, sealing all the cells from the outside, and then pumping water in to form a cavity inside with fluid. In this case, it's programmed cell death. That there's a bunch of cells here, and some of them are killed off to make room for the expansion of this cavity. This is the amniotic cavity. Eventually, this is going to have to cover and surround the outside of the embryo. That's how the embryo is protected, cushioned inside the mother, from kept in a nice moist environment and so on. But for the moment, it starts kind of in the middle, and programmed cell death is one of the ways that this occurs. This is the epiblast here, single sheet of cells. There's about 1,000 cells just in the epiblast at this point. So this is by no means accurate. Remember this, this is a cup shape, so we're looking at a slice through it. And I think that's probably it. If we stop here, so this is, we're just about to start gastrulation, and a primitive streak is going to form, and it starts kind of here, and then, and it's not a groove the way it is in chick. It's more of a thickening. So you can see what looks like a streak, but it's because the cells get thicker. And that is what we will talk about in lecture 20.